Hello and welcome to the Hosted Datasets in the Ecosystem video module, which is a part of the 7 Bridges onboarding lesson. While watching this video, please keep in mind 3 key concepts. The first is that the Cancer Genomics Cloud hosts the data in the ecosystem in cloud buckets and supports all the data storage costs. Because the study data is not moved, but only symbolically linked, researchers do not need to spend time moving data or paying additional storage or egress costs. Finally, there's a wide range of data available to researchers that includes TCGA, TCIA, as well as other important datasets. At the end of this module, you will be able to understand the depth and breadth of the hosted datasets available to you and understand how data access works in the ecosystem. Let's start by talking about how datasets are hosted on the CGC. The CGC ingests the research data and puts it into Google Cloud and AWS buckets that they own. The data is then put into a file management system and made accessible to an authorization service. Access is programmatically controlled by the authorization service. When a researcher chooses to access a particular piece of data, their approval status is read from dbGaP programmatically. The same data is hosted on Amazon Web Services as well as Google Cloud Platform and is available on the Cancer Genomics Cloud Platform. Let's talk about the types of data that are available. Some of the categories of data include genotype, proteome, or phosphoproteome, and imaging data. Popular formats for each of these data categories are listed in this table. Some of the data is controlled as mentioned earlier. This means the platform will programmatically check your dbGaP credentials to make sure that you have the appropriate access levels. The controlled access tier includes information which is unique to an individual. This includes most raw data files and some processed data, such as primary sequencing data, both in BAM and FASTQ file format, from DNA, RNA, microRNA, or bisulfite sequencing studies, raw and processed SNP array data, raw and processed exon array data, and somatic and germline mutation calls for an individual. The CGC also has a lot of open access data. This is great for students to have something they can use while learning cloud computing and analysis. Open data is also great for testing tools and workflows using different types of studies, as well as benchmarking your analysis before scaling up to your full study. Important open access datasets include the Cancer Genome Atlas, which features over 500,000 files spanning 27 disease types, and the Cancer Institute's Clinical Proteomic Tumor Analysis Consortium, or CPTAC. Here you'll find over 60,000 proteomic and genomic files associated with over 1,000 cases. On the CGC, powered by 7 Bridges, you can use the data browser to find open as well as controlled datasets. The platform will let you access only the files for what you have the appropriate approvals to use. But it's also a great way to see all the breadth and depth of all the different studies that are hosted on the CGC. I hope you now have an understanding of the datasets, and I want you to take away these 3 points. The first is that the Cancer Genomics Cloud hosts a wide range of datasets, some of which require appropriate dbGaP permissions to access. Because the data is linked to cloud buckets, researchers do not have to pay for raw data storage costs. And lastly, a wide range of data is available to researchers, including TCGA, Target, TCIA, and open training datasets. That concludes this video. The next suggested video is called Find Hosted Study Files with Data Browser. This will go into detail about how to use the platform to define open as well as controlled data and link them to your workspace.